Oh yeah? Do I even need this?
That microphone is for the recording.
Oh,
And
thank
for
you.
people joining us on Zoom.
I hope you haven't. I hope you haven't recorded that.
We'll see.
Okay. So thanks for the nice introduction, Jim. I want to make this as informal as possible. I prefer if you have questions, ask them when the slide is up. I think that's better. I have this little cheat thingy here. I will make sure that I don't use much more than 45 minutes, including the questions. So I will drive it forward if there's too many questions. I've also prepared a talk about fertilizer, all the background, the science behind it, that I can give at any time if there's interest. I figured it's too much to pack all into one. I would be talking here past noon, and I think you will be half asleep here. Your interest will drop. So I thought I present this first, and then the theory behind it. The other way around would probably been a little bit better, but let's start it this way. So let's put this here. And yeah, 14.2. Okay. So the question is, what does fertilizer do to carnivorous plants? And I focused in this experiment on the Sarracenias. I got some seedlings from Jim, as he mentioned, and I paraphrased Chris as fertilizer being napalm for CP roots because I couldn't find an as good analogy of what you read in the literature. I don't know if you guys know what napalm is and how it works. And I can tell you up front, it's not napalm. It has nothing, it doesn't do anything close to what napalm does when you drop the bombs. So, yeah, it's pretty gnarly. So why the hell would I even do this? And so the reason is basically the napalm interrupts. But I grew carnivorous plants started in 2000. And I had a few Sarracenias, I even had Darlingtonia, a few Nepenthes, and I managed to kill them over the next 22 or 3 years. And then in 23, when I wiped out my last Nepenthes ventricosa, which was a ginormous thing that had vined all over my greenhouse, I got back into it, first into the Nepenthes, and now the selection is way better. But I thought, let's not going to do the same mistake and actually read up. Back when I started, it was actually good. There was only one source, and that was Peter Damati's book on carnivorous, booklet on carnivorous plants. There was basically, the internet was in its infancy. So all the information I got was this booklet. So I thought, let's do better. And I did a lot of reading. And to summarize it, all the websites warn against using fertilizer on Sarracenia and basically on any other carnivorous plants. So I found one article, however, that contradicted this. This was an International Carnivorous Plant Society newsletter. And then they made a part of their website based on that article. It's John something or another. I forgot his last name. Can I make this a little bit smaller because the text is cut? Do you adjust this? Nope, the other way. Oh no.
Let's focus.
Yeah, this is focus. Do you have to move the table closer? Oh, there it is. Yeah. Yeah. And make it smaller. Sorry. Is it better for this to present okay, because I could also save the presentations in a 4:3 format. This is 16:9. Okay. All right. So this article kind of contradicted this, and he showed a picture with a clear positive effect of slow release fertilizer on some Sarracenia seedlings. The highest amount had the greatest effect. He used, but he didn't go all the way to see a negative effect. That's what I like to see, so that you have when I do experiments, you want to get an optimum curve, so you want to know that that was the optimum. So this is a bit inconclusive, and he also states, but doesn't show any data, that fertilizer basically doesn't do anything to adult Sarracenias, which make me scratch my head a little bit. But so this is all what I really found as hobby information. Is I don't know if these people are just writing and copying from the previous person, but it's pretty much the same information. So commercial growers, I've heard rumors that they do fertilize the Sarracenias to produce them fast enough, but there's virtually nobody reveals anything because it's obviously a trade secret because you don't want to let your competition know how you do it. And then I turned, because I'm a scientist, I returned to the scientific literature. Not that I say that this data is any better, but what I like in scientific literature that is clearly documented what the people did. The methods are there, duration, it's all written down. So the hobby information is sometimes you read about fertilizer, oh, we fertilized something for half a year and then it died on us. It has to be the fertilizer. So that kind of information really doesn't tell you anything because it lacks information. What happened when your plants died? What fertilizer did you use? What were the conditions? If this is all described in the scientific literature, that's why I prefer it. And to say the least, I have one slide on it. It's conflicting, I put it mildly. And when you look at all the carnivorous plants, it gets even more conflicted. It's really, I couldn't make any rhyme or reason out of the data. It's very confusing. So I said, hey, why not? Let's do my own testing. And don't think that this is a complete finished thing. I see that as an experimental set, like I would do in the lab. One experiment triggers the next experiment, and so on. But I answered the first questions I had. So before I go into my presentation, I set the introductory slide. So there's some very basic stuff on plant nutrition to refresh everybody's memory. A synopsis of the key data from publications, and I can skip this. Oh, and this thing, actually, I broke it. It doesn't move. I spent 5 minutes, so I have to do it this way. A synopsis of the key data from publications. I made it as short and concise as possible. And then something that's near and dear to my heart, I will have one slide on TDS versus EC, and you will see, hopefully when we get to the slide, understand why. No, don't tell me this thing also died.
No.
Oh, yeah. So plant introduction. You guys all know that plant nutrition, the nutrients are divided into two general groups, the macronutrients and the micronutrients. And this is basically on the fact how much a plant needs. So macronutrients are usually measured in ppm, parts per million, whereas micronutrients are measured typically in parts per billion. So there's a 1,000-fold difference in the requirements of the plants. So plants are, obviously, we know 95% water, so it's not surprising that the main molecules plants are made of, carbon, hydrogen, oxygen, we don't have to worry about. They come from water and the air. So there's no need to fertilize, although you probably know that some marijuana growers use additional carbon dioxide because it's actually rate limiting, even with the current elevated levels. I can get into this a little later. So what you all know, NPK are the main components. So N is probably the most required nutrient. Everybody knows that. So it's used to make macromolecules. DNA contained it, protein contained it, chlorophyll contains it. Without this, there is no growth. You can't make DNA, your cells can't divide, you're done. What plants can do, and I will get to this here, is they can mobilize nutrients from the rhizome or from old pitchers and old leaves to fuel new leaf growth. And that's what they typically do in the old parts, then die off. That's called senescence. You don't want to get there. You want to maintain the most plant material you can. So you want to supply these. Phosphorus is key to energy metabolism. You all have heard of ATP, adenosine triphosphate, or creatine phosphate. Without P, there is no energy metabolism. There's no photosynthesis. You cannot convert the energy the plant collects into making material and store the energy. So P is absolutely essential, and it's also part of some macromolecules. So for example, DNA. DNA is made of adenosine triphosphate, phosphate, right in the name there. And then interesting, the third big nutrient is potassium. Potassium is interesting because it is not incorporated, unlike most of the other nutrients, which are parts of enzymes or actually part of structures themselves. K is kept in the plant and it maintains turgor pressure. It's a little bit analogous to our salts in the blood. That's all we need to know. And the typical macronutrient requirement, and now it's key. So Plants need the most N and K, then followed by calcium, then followed by potassium, phosphorus, which is about equal to magnesium. And that's the two things that many carnivorous plants growers forget. If you use RO water, you take out the calcium and magnesium from San Diego tap water. You need to resupply calcium and magnesium in a soluble form. Without this, your plants will be deficient, and that's why I have these two in bold. And calcium is very critical. You might know that. It's incorporated in the cell wall, structural rigidity. It's also a critical signaling molecule, both in vertebrae as well as in plants. Without calcium, there's no signaling. This is very important, for example, in stress and pest response. Magnesium, you all know, it's a catalytic ion. Many enzymes use it, so it's at the center of chlorophyll. Without magnesium, chlorophyll can't convert light into usable energy. Many other enzymes, for example, polymerases, DNA polymerases, use magnesium. Without magnesium, they're not active. They can't polymerize, so they can't make DNA, they can't replicate, So is this kind of clear where this stuff goes? And the micronutrients are minor components of good fertilizers. In soil, they're generally not deficient because our soil here has tons of these things. They typically are deficient because the soil pH gets too high. And this is not hypothetical because it's actually been shown in agriculture, also in hobby plants. So palms are and cycads are very sensitive to manganese deficiency. And that's not because manganese is not in the ground. It's because when you irrigate with tap water, the pH drifts up and manganese becomes unavailable to the plant. And the order of what the plants need, of course, iron is the biggest one, manganese, zinc, copper, and a very little boron and molybdenum. So as I hinted, there is some big distinction in the nutrients, whether they're mobile or immobile. Immobile means that the nutrients cannot be transported. They cannot go from an old leaf to a new leaf, which is very important. So calcium is one of those. Almost all micronutrients are like this, except for these two. That means two things. First of all, if you have deficiency in an immobile nutrient, you will see it on the new growth. With the mobile nutrient, you will see it on the old growth because it gets mobilized out of the old growth. So if your old leaves get green, then it's a nitrogen or iron nitrogen deficiency most likely because iron is immobile. And if it's the other way around, it's more likely to be iron. So kind of it helps you to, if you see deficiency, to figure out where they potentially come from. The other thing is also the immobile units are easy to cause deficiencies because easier than the others because you cannot mobilize them from old tissues. So that's why calcium, especially if you use RO water, is one of the prime candidates for deficiencies. Oh, that's why it doesn't work. I need to point. Oh, I kind of blocked the, you know what? Since this is not working, I can take this out of the way. So one last thing to nutrition, and that's an important thing. It's Liebig's law of the minimum. It's kind of obvious to us today, maybe, that plant growth is not guided by a complex accumulative formula. It's limited by the least abundant nutrients. And I like this depiction. It's always depicted as these barrel, and you can see the little wooden things here have different heights. And here it's depicted as phosphorus being limited. So that kind of means you can put as much nitrogen into this, it won't do anything because phosphorus is limited. And of course, it shows other things than just a fertilizer. So all the environmental conditions and a fertilizer components have to be optimized to optimize your growth. Otherwise, you limit it on a certain limiting factor. It is interesting, so it seems obvious, but Justus van Liebig published this in the mid-1800s. This is when America was in the Civil War, to give you a perspective. Plant nutrition back in the days wasn't a strong field. Very little was known what plants really required was, for example, not known that CO2 comes from the air at the time point. So it's that puts it into perspective. Interesting is I mentioned CO2. So under normal highlight, well-fertilized plants, CO2 becomes the rate limiting factor. Even the 400 ppm we have in the air is below the maximum. The maximum is anywhere between 11 and 12 to 1300 for plants. So 3 times the current level. That's why marijuana growers, they're grown tents or rooms, they pump it full of CO2 because they give the plants incredibly strong light, huge amounts of fertilizer, really huge amount of fertilizer. And at that point, CO2 becomes limiting, and they want to eke out every dollar out of every square foot they can. So for them, it makes sense to actually get a CO2 system with pressurized tanks and regulators and all this, and they pump their rooms up to about 1200 ppm. And it's unexpected if you grow your plants outdoors almost, and it's otherwise well-balanced. CO2 is limiting. Should I actually skip this? There's a lot of text. And it's very complicated. I only want to mention one. So some of the very early studies, I'm not sure where these guys got the horticulture degrees. They did plots of Sarracenia and they dumped a shitload, literally, of synthetic inorganic fertilizer on it. I did a back of the envelope calculation. It was 10,000 times the amount I applied to my orchids. And for my orchids, I used 10 times the amount for Sarracenia. So I'm actually surprised that the Sarracenia survived this, but they obviously weren't happy. And their statement is that fertilizer is bad for Sarracenias. And that's one of the, everything can become toxic to anything, right? It's a matter of quantity. So, and I think that has reverberated up to this point, that it's still somewhere in people's mind. These experiments were really crazy. There's a number of other interesting findings. I skipped this so that we can go through the experiment. So TDS, and Jim Bowman alluded to that. So there's EC and TDS are 2 different things. TDS only applies to an evaporated volume of liquid, and you measure the grams that come out of this, and it's expressed as ppm, which is usually milligram per liter. So this is a physical weight measurement after you dry stuff down. Nothing else is TDS. What people typically refer to as TDS is a measured thing with these little meters, which are wrongly called TDS meters. They are actually EC meters. EC measures the electric conductivity between 2 little electrodes in a liquid. So if there are no electrodes, so absolutely pure water has 0 conductivity because there's no ions to flow. And I think TDS is more popular because people, I don't think, like the units, which is micromillisiemens per centimeter. It's probably too complicated to pronounce. So why does it matter, and why should we use EC? So the TDS meters do not measure TDS, they measure EC, and they should be called EC meters. They convert that, though, based on a scale. That scale applies to only the specific nutrient or ion. The scale that's typically used is sodium chloride as a reference. So if you measure salt water, that's perfect. EC and TDS translate in a 1:2 ratio. For any other salt solution, that equation falls apart, and the measured EC cannot be translated into TDS anymore because it is specific for that calibration. So a good example is, to make that clear, the fertilizer I used in this study, I used 70 grams per liter for 100x solution. So that's 0.5 grams per liter, 700 milligrams, so that's 700 ppm. When I measure this, and this recommended solution in the fertilizer sheet is about 800 microsiemens per centimeter, and that's what I typically measure around that. So every time I make a fertilizer batch, I measure the EC to make sure that I didn't somehow introduce an error, which the meter, of course, converts to 200 ppm. So you have 700 actual ppm versus 400 reported ppm. So that's a 1.75 fold difference. And that's why it's important, and any nutrient, anything, you say which fertilizer it is and what the EC is. Because another fertilizer will have an entire different ratio again. So it is really specific for the solution you have. And this, I think it's kind of important, because if you report numbers, you want to report proper numbers. That's the point of having numbers and not eyeball things. Is that kind of, does that make sense? Okay. So the goal of my experiment was, and I would always recommend it for any type of experiment, make it as simple as possible. I find that best in my career as a molecular biologist. If you want to answer too many questions and make it too complex, often in the end you end up with 0 questions answered. So it's best to go slow, ask one question, then do a follow-up experiment and ask the next question. So that's why I did. The other thing that is also why I had to do it, because it gets quite complex with plants. You need a lot of replicates for it to see if that's reproducible. I can't do a complex experiment. It's just I don't have the capabilities and the space. Only commercial growers could do that because they have greenhouses full of similar plants. So I asked 3 basic questions. First, based on the napalm phrase, do Sarracenias even tolerate watering with inorganic fertilizer? So this was my main objective. Will we damage them, especially the roots, or not? Second, can they use inorganic fertilizer? So basically, does it help them to grow? And third, where would be a good concentration? So is more better? Not always is more better. And I have for like over 2 years now, I used my orchid fertilizer 10-fold diluted. And all my CPs, I have not seen any detrimental effects. And I've tested roughly no ROE only and 10% ppm. And I did not kill any plants that way. Not specifically in the fertilizer group. I've killed CPs, but independent of this. So I decided, since I already know that this is kind of tolerated, I went with 20, twice as much, 50, 5 times as much, and then the 100, which I use for my orchids. And the 100 was specifically, I only had a few extra plants from Jim that were kind of bigger. So I said they go in a 100 ppm group because I was figuring with the bigger plants, I see stress easier and they're also a bit more robust. So this was not an experimental group where I only compare 20 and 50. The 100 was the stress test. So experimental setup, I can go through this. It's a lot of tests, but I can go through it quickly. Plants came from Jim, as I mentioned. I acclimated them. Then this was a compote. I potted every little seedling individual in either 2.5 or 3.5 pots. The 275 is shown here. The medium was peat, perlite, coarse sand, the typical Sarracenia medium that most people use. Sunshine was the manufacturer or the source of the peat. Then I let the plants recover, and then I started the experiment. So I started in September. I already mentioned I had 2 main groups, 24, 50, and 2 sacrificial plants at 100, expecting to see shit happening at 100 ppm. The plants were kept indoors because by the time I get everything ready, September is, I don't think, a good time to start an experiment outdoors, slowly going into winter. So they were kept indoors under LED lights with 16-hour lights. And they were initially kept in 1020 trays with a humidity dome. And then you will see next I have pictures initially. And then I actually, luckily, I used to breed reptiles, monitor lizards, chameleons, poison frogs. I had still cages sitting in my garage. So I very quickly had to recruit those terrariums because my Sarracenias outgrew the 1020 trays and domes. So that was a bit unexpected. The temperatures, my garage is fairly warm. I have these aluminum doors with glass windows. So especially in winter when the sun is low, it can heat the room up into the 80s very quickly. And at night, since there is occupied space above and two sides, the temperatures don't drop much, and I have a little space heater in there that if we get a really cold night, keeps it at about 68, 65 or so. So, so they should go through winter on this constant thing, not knowing that there is winter. That's the point of it. And watering, I did something a little different than how most people water the Sarracenias. I don't have them in standing water. I put them in these little cups, added the pot to it, and then added a specific amount of fertilizer solution, either 100 mils or 150 mils for the bigger pots. I let the pot sit 15 minutes, and I poured the liquid back and forth. I moved the pot to a new cup, poured the liquid, and went back and forth so that I equilibrate the solution in the pot, because I was very, very afraid with this fertilizer that I have salt buildup. And my question was not, do I get salt buildup? My question was, what does a precise dose of fertilizer do? So I thought that this was a necessity, and you will see in a minute that it actually was a good idea that I did this. It costs time. So to water the plants for this experiment cost me about an hour and a half every Friday. So towards the end, it was a little bit of a pain in the ass, I have to say, but it's much easier to have a tray where you pour your water in than doing this. Plus, you have to handle all the plants. You have to take them out of the terrarium, out of the things. It's extra work, but I think, my opinion, it was worth it. So the key is no salt buildup. So the plants is also important, different than what you would get outdoors. They get nothing else. There's no insects in my garage, at least that I know of, that could get into the pitchers. And I didn't fertilize or water the pitchers itself. So this was all only root zone. Because the reason is KISS, right? I want to keep it simple. I have many other questions with how there's an interaction between root and pitcher, but that would have made this experiment way too complicated. So, and they were not sitting in trays, as I already mentioned. Sorry. I think this thing is, okay. So this is a picture of my initial setup. So here you get the size of the plants, and this is the plants now, and I brought a few in. So here, for example, so these are the aladas here. They were, and I potted them based on size. So bigger plants got into 3.5-inch pots. Smaller plants were in 2.75-inch pots. You see that here. And they were kept in these trays, so either 1020 or 1010, and then I used a 7-inch humidity door, a dome over it. The lights were initially the 4-foot Flora wave or iron lights. Those are common LED lights that Carnivorous sells Flora wave, and a local shop sells the iron light. So this is in late September to mid-October. And you can see this is the dome, and this is the tray. I had to, the Sarracenias were outgrowing this tray. And I was lucky I found these insertions on Etsy. So I bought them. I bought 5 insertions. So at least it bought me some time. I also had to reactivate my terrariums and get lights for them. And so this was a terrarium that I initially had young tree monitor lizards in that was now housing Nepenthes. So I had to kick the Nepenthes out into my warm house, and I started populating the tank with Sarracenias that you can see here. And again, I didn't plan on this, and I rather would have not liked to do this, but this is kind of like shit what I'm doing now. So I had to come up with something creative. This is towards the end of January, and now you see the terrarium. If I go back, yeah. So you can see where the Sarracenias are level-wise. They're kind of down here. Man, this is, I don't know. And now look at this. So now the Sarracenias are way taller. You can see some of them reached quite up into the terrarium. And I had to, on the humidity domes, I had to add a second extension. So this was all I could fit under my wire shelves. Otherwise, I would have to rearrange the entire wire shelves in their storage boxes. And now this would have been a royal pain. You can also see the floor wave lights. And you can see that a lot of the Sarracenias are already basically hitting the top. And this is at the conclusion of the experiment. That was mid-March, so now the 6 months. I plan to do the experiment for 6 months, and that's all I wanted to do because I was. It was ended up being a bit of work, the watering, and you can see there is the last group of kind of stragglers still in the humidity domes. That's all that's left. These plants here, and you can see these are the ornatas. Flava need to be moved here. And in the back, you see a couple smaller stragglers. Those are the last ones. And they are still stragglers. They have outgrown this now, but barely. So there is diversity in the growth. But you can see how tall they are now here.
Are less 2 years old, I think.
Jim, when did you seed them out? So they got fertilizer under my regime for 6 months, right?
They were probably sown December 23.
Yeah, 2 years then. Actually less because it's December. This is March, so 1.5 years. So I mentioned to you my fear of salt accumulation. So I really measured the efflux, the pH, and all of this. And as you can see at pH at 20 ppm, the input is about 180 microsiemens per centimeter. So for those TDS people who can never convert, it's about 90 ppm. So it's below the 100. And then, of course, with 50, it's 2.5 times 380. And at 100, I mentioned that this fertilizer is about 800 microsiemens per centimeter. When I measured the efflux, it was interesting that the 20 ppm consistently over the entire experiment came out slightly below the input. I cannot tell you where the nutrients went, whether they were bound by the peat. The plants took them up and converted them, but the key is there was no accumulation. With 50, you can see a very slight accumulation. We ended up with about 10 to 20% higher. However, the 100 ppm group, you can see that after about 4 months, we exceeded 1,000. And then at 1,200, which is 150 times, I initially had planned to run the experiment until the plants die. But at 1,200, I chickened out, which was also 150 ppm. So I flushed these out and readjusted them to 50 ppm because by that time they looked really nice. And I'm a hobbyist. If that would be a scientific experiment for a journal, I would have drove them into the ground, but I decided not to do that. Yeah, so here is the root system. So I think whenever you start an experiment like this, you want to make sure your plant has a healthy root system. There is no point in dumping fertilizer on this Nepenthes import that has one root that's the thickness of my hair. There's nothing to take stuff up. You have to be lucky if that plant doesn't dehydrate. Luckily, Jim's plant had a phenomenal root system. So they really had you can see a healthy root system. I unpotted them, I completely cleaned them out, and then potted them in my mix so that they're all in the same mix, same pots. So this was really a good start. And you can see at 4 months, and why 4 months? I decided that these plants were in 275 pots. They became too small, so I wanted to uppot them in 3.5. And I just did that for a few plants, and I showed 2 here. I wasn't as careful with removing all of the mix because they went back into the same mix, but just to show you the root system. So it's not like I killed off all the roots. And this is a 20 ppm plant, and this is a 50 ppm plant. So root system at that point, after 4 months, was perfectly intact. So this is at the end of the experiment. So this is all the alatas, the 20 and the 50 ppm group. So it's 4 plants here and 5 plants here. And you can see the root system is somewhat variable, but it's good in general. You can see, I mean, this one has humongous root systems. Some of them grew out of the pot, so I actually had to cut the bottoms off. So, and you can maybe argue, well, this one is kind of, it's a big plant, but there's not that much in roots. So there's some variability, but I wouldn't say there is much of a difference between the two groups. And for certain, I didn't napalm these plants, right? Still alive, still have roots. And this is the 100 ppm two flowers, and you can see the roots grow out of the pot. So they also had a healthy root system. This is the 2-month time point. And I think I have another photo. Yeah. This is the end of the experiment. Remember, 4 months, 100 ppm, then down to 50 ppm, but you can see the roots look good. They come out of the pot. Also at the end of the experiment, I repotted everything, took it out, washed it, measured it, and you'll see the measurement in a minute. Root system looks really good. And this is 100 ppm, which I wouldn't recommend because of the accumulation I saw. But this is what I use in my on my orchids. And this is also 100 ppm is kind of the standard in the horticultural industry. Okay, so conclusion one, I was a bit overwhelmed by the speed that these guys grew at, and I had to reconfigure my system a couple of times. You normally don't want to do that during an experiment. You want to keep the condition, but it was impossible. Now I know. And I can say clearly that Sarracenias can tolerate inorganic fertilizer, even at 100 ppm for that time period. And remember, I kept them long enough. This would almost be a full growth season outdoors, because I wouldn't throw fertilizer on dormant plants because they're not growing. What's the point? So this covered almost the entire growth season if that were be outdoors. And then now the question is, well, is there a benefit of going higher? So one thing that I noticed, and it's obviously, it seems like common sense, that this one is the fold growth increase and this one is the initial pitcher height. So there's an inverse correlation, which you would expect. A pitcher that's already 10 centimeters tall can grow 20-fold, won't be this tall. There's a physiologic limit of what plants can do. So you get this inverse relationship. And that, of course, makes the data a little bit harder to interpret because you have plants at different heights. They're not all exact clones. And then you want to see the difference in growth, but you start out with different heights that then infer different growth. So when I saw this, I was like, oh boy, I wasn't expecting to see anything because of that variable and variability on top of everything. But this is what I saw at the end of the experiment. The two 100 ppm Sarracenia plants were those plants here, right? I picked them because they were bigger than everybody else, and this was not part really of the experiment. And because of this correlation, you see they actually apparently grew less, but this has, I don't think, anything to do with the fertilizer. This has to do because the plants were bigger, and that's why I kind of excluded them from the real main experiment, but I want to show it to you because it's part, I showed you the root system, so I want to show you all the data. And this is all the other guys. So you can see the fold size increase is very variable, but overall, you got a little bit more with 50 ppm than with 20. But given the variability, and that was basically haunts the scientific literature with carnivorous plants, a lot of the studies see differences, but they're not statistically significant. This is really the pitfall of any biology with something live. I'm a biochemist. If I do an assay twice, it better give me the same data, right? With live things, especially if they're not cloned, they can go any way unexpected. So I was like, oh boy, this doesn't look, I'm not going to get anything statistically out of this. So, but this is a table of the parameters. So the initial height is identical between the group. I kind of grouped them so that they would be, the height of the end of the experiment was higher in the 50 ppm. 28.6 was a 38.2. And this was actually indeed statistically significant by t-test. So I was like, wow, I was positively surprised. And so is the height gain. Height gain is this height minus that height. It's also statistically significant. The percent increase, although goes from 676 to 917, there's a clear difference. But because of the variability, this was not statistically significant. And that's what you always get. You have to be very, very careful with live things, as I mentioned. So I was happy about these, Tim. The number of growth points in 6 months, I wouldn't expect. So I measured the number of different growth points, was about just under 4. And the weight, so I unpotted them, weighed them all, is slightly higher with 50, but also not statistically significant. So, and of course, this is a hobby study. Yeah.
Standard, your point, 0.05.
Yeah.
Okay.
Yeah. I mean, you could go, there is a complex statistical tree of where you have to put this. I typically go with by the 0.5. This is generally accepted as a, I mean, I'm not publishing this and not publishing this. So this is a hobby study, right? So these are very easy parameters. In the scientific literature, typically at the end, you destroy your plants. You do dry organ weight. You can do all these parameters here, measure the photopigments and relative water constant. I mean, there's a million of things that you could do, but they require destruction of the plants and they require probably multi-million-dollar equipment, gas chromatographs, mass specs, and the whole nine. I don't have that. So this was out of the question. I wanted to keep the plant, especially since I took so much effort in raising them. So one of the things that is in the literature is that fertilizer triggered phyllodia. So I'm showing you one plant, and this is actually the flava on the leftmost, next to, yeah. You can see it has some phylodias. So what I found was actually interesting. None of my plants had phyllodia until I chickened out on the 100 ppm and washed them out with RO and switched them down to 50 ppm. And the next growth of leaves was phyllodia. And you can see them here. So I only 3 plants developed phyllodia. That one here, the other plant that was also in the 100 ppm developed phyllodia. Fewer, and then one 50 ppm group, but only after the experiment, and they were actually now sitting in a tray and watered with 20 ppm in. So every time plants developed phyllodia, it was after I reduced the fertilizer, not doing the high fertilizer regime. I found that curious, but I just wanted to show you that. And it's more, so none of the 20 ppm plants showed any phyllodia. So there is a correlation that is happening after reduced fertilizer. God knows why.
So you think it'd be the opposite?
Correct. It wasn't. I don't know. I'm almost done here. If this thing will ever go to the next slide. Final conclusion. See, I'm almost done. So it was a lot of work, but it was also super fun. It really, and I learned a lot how to handle Sarracenia seed. I always recommend everybody to start growing stuff from seed. You learn much better how to grow things. Plus, you see the variability. And you can cherry pick the plants that you like the best. If you buy something from someone, most of my big Sarracenia are bought, and I get everything from something that looks really good to some runts, right? There's whatever they find, they throw in a box and ship it to you. Here you have a control. You pick whatever you like. And you also have to see that I got those compounds from Jim for $15, and they contain 4 to 7 plants. I mean, can you complain? That's just like $2 per plant, right? And a little bit of my time, a little bit water and fertilizer. And now you have these big plants that you see in the back there. I already mentioned that Sarracenias grow way faster than I anticipated. They can tolerate soil, inorganic soil fertilization, even up to 100 ppm for at least 4 months. And even with that salt accumulation I showed you, And I would say not only can they tolerate inorganic fertilizer, but they benefit from it. So they really grow faster. And I was surprised that I got the statistical significant results as I showed you. So this is now in June. I took a picture of the same terrarium. And most of the plants now have to go into my greenhouse because I don't have a taller terrarium. But you can see here the Supermax flowers and a number of the aladas are actually going to hit the glass ceiling. You also see that some of the pictures have a little bit of a burn. It turns out that the lights, it's getting really, really hot there. And initially I had the lights sitting on the glass. That was not a good idea. So I now suspended it about an inch. I had to drill holes in my ceiling and lift it up. And I have and you can kind of see that I have a little fan set here, a little computer fan that kind of blows the heat away. It got too hot up there and some of the pictures shriveled a little. And one thing, yeah, you can see something like here. And this one, I actually had one allowed to bloom, which surprised me because there's no winter cue in there. They shouldn't really start blooming, but so that means they're mature. And this one isn't one of the biggest. You can see it in this picture. This is the flower here. So you can see there's aladas that are easily twice the size. And I didn't bring the aladas. I bought some Supermaxes and some ornatas. So, and I left all the labels in there with the ppm, so you can look at the plants. So what I just noticed last night when I was taking the plants out to bring here, there was actually a difference between 20 and 50 ppm in the aladas. So I tried to capture that, not a very good photo, but I grouped the 20 ppms on the left side and the 50 ppms on the right side. And you can kind of see here are the pictures and here are the pictures. So there's a clear, even though for the last 3 months they were all getting 20 ppm, they still had a memory, stored energy, extra stored energy, probably in the rhizome, to get you this enhanced growth here. And these are basically as big as the mature aladas I have up outdoors. They're full grown. The terrarium is 36 inches tall, minus, of course, the pot, and minus there is a little bit. I have a pumice in the bottom and the tray, so there's this much. So about 6 inches occupied space in the bottom, and they're about 3 inches or so away from the top, so 24 to 27 inches tall. So I have a couple recommendations for the group. You know, don't listen to what I say, do your own experiments. That's what I always tell to everybody. When I came as a grad student to the United States as a postdoc, my postdoctoral advisor completely floored me. I was a naive grad student, scientific literature that was set in stone, that was the Bible, peer-reviewed, always correct. And he's like, don't believe anything in the literature. I was like, what the fuck? So and I learned that over my career, over the 30 years afterwards, don't believe anything in literature. Don't believe anything you read, do your own experiments. If you make divisions, take your divisions to do experiments like this on your own, under your conditions. You know, these are my conditions that may not apply to you. So your own data is always the best data in my experience, because you know the pitfalls, you have generated it, you know everything what you did in the experiment. And when you do that, write it down, because you'll forget. Document everything. So I mentioned I tested all my CPs up to 10 to 20 ppm. I have not seen any adverse effects. So you won't blow apart your CP collection by testing moderate amounts of fertilizer. Here's actually something I just took a couple of days ago. I bought a Drosera regia from Chris, and I read up on this, and I was surprised to find that they recommend fertilization for this. But then the author kind of made a joke and said, but do not get it on the other Drosera around your regia. I won't be responsible for you killing your Drosera capensis. And I was like, absolutely perfect. I have a lot of capensis to kill. Let's get going. So I did this in March. after buying your regular, a few days, I repotted 3 Drosera cup, and they were exactly the same size. They got into the same mix, same pot. And ever since then, and they are in trays, I top watered them until the tray mildly flows up. I use a distinct volume, 50 ml early, and now I use 100 ml because it's now hot. And you can see this is RO 10 and 20. So it's definitely not, these two were not treated with napalm. They're there and they're looking good. There's 1 flower spike here, 2 here, and actually 3 here. And I would say, I'm not claiming that there's a benefit here, but to me, this plant looks bigger than that plant, and that plant looks bigger than that plant. Oh, yeah. Yeah, the sphagnum also likes the fertilizer. I mean, you can see the Sarracenias in there. This sphagnum is 3 months old. Yeah, it grows like, and I ripped out most of the sphagnum and left it at home. So while saying this, I haven't had time to do detailed studies with all the other groups. I can just make this general statement. So when you do your own experiments, so first, before you start, you should optimize your conditions. If you probably already have your own optimized conditions, make sure that you don't have to change them when you do the experiment. Try to plan ahead. Don't, and the one thing that is important, never apply something new to your entire collection. Even the fertilizer manufacturers specifically state that. Take a few plants, fertilize them first, and start with a low dose and then escalate to see how your particular crop responds. You can imagine what commercial nurseries could lose if they fertilize like 10 full greenhouses and everything dies. I mean, they're bankrupt, right? I would also recommend that for you, just start low. If you make division, take one of the divisions, treat it, the other one not, and you see the difference, right? At least you can rule out toxicity. You can even do an escalating plant on a single thing. You won't see differences, but if you just looking at toxicity, you should be able to discern, oh, now the plant's going downhill. Ideally, you stop before it goes downhill, so you kind of see, oh, not doing that well, so then you back off. And I am always, I mentioned that already with the TDS, I'm a big fan of measuring. Measuring and get numbers, and not just, oh, this was high, low, water a little bit, weekly, like the orchid community did. No, you water with a specific amount of ppmn of a specific fertilizer. You write that down. You want to be precise, because measurements are useless if there's no precision. You have to be relatively precise. So when I weigh out fertilizer, I use a scale. And then if I do it lots of time, the same fertilizer, I mark a cup. And this is about what I need. But then I go back every time I have a solution, I measure the EC. Make sure it is what it's supposed to be. Because you can very easily make a tenfold or a comma mistakes on Excel, and then your CPs, if it's on the high side, will probably suffer if you dose them with 1,000 ppm, and I won't guarantee that they survive that and like that. So
You use just a nitrogen only fertilizer.
what?
It was nitrogen only.
No. I get to this in the next slide. Good segue. So measure also the efflux. So especially if you use a table like Sarracenias and you just bottom fill, the nutrients will accumulate unless the plants take it up, right? You have evaporation and transpiration of the plant. So if you keep on adding nutrients, you will add more and more and more. So you have to measure your efflux. That's why I did this complicated going over 3 times. If you grow them in a tray, you have to measure what's in the tray. And then if it gets too high, you have to flush everything with RO and reset it. So you have to keep an eye on this. That's absolutely important. And I think that's what happens in the literature, that people don't pay attention and dump fertilizer on it. You get salt accumulation. And the other thing is, and I'm going to get into this as my fertilizer presentation, fertilizer has an effect on pH, every fertilizer, and it varies on what's in there. So it's actually relatively complicated. The interaction of fertilizer and soil can be extremely complicated. And I bet you these guys didn't pay attention to pH, and a lot of the carnivorous plants that grow in acidic conditions are sensitive to pH. They want to have a specific, relatively acidic pH, but they don't want to be sitting in pure acid like pH 3. They want to be around 4.5. So, and they don't want to sit at 7 either. So, and I bet you this is what it's in the literature when you see this, that people do not pay attention. So you have to pay attention. And in my main talk, I go into the equipment I use. It's very inexpensive now. You can buy relatively high quality material. So yeah, KISS, one question at a time. When you do a group, try to have a control group and try to have at least 3 to 5 plants per group so that you can compare, because plants will be variable. And here again, take good notes and photo to document, because I've done so many things to my orchids and not written it down. Like 20 years later, I was like, yeah, I did this, but so write it down and then you have it forever, unless you lose the computer file, which happened to me. So and allow time. So fertilizer, it's not like you apply it and boom, it changes everything. So there will always be a delay. It's probably, I would say, in a month time frame. So you have to be prepared to do an experiment, I say, for at least 6 months. So recommendation 3, and this is where I get into the fertilizer. So I used a fertilizer that was developed by Michigan State University for pure water. The only thing that this fertilizer is different than the others, it supplies calcium and magnesium, because you take that out with RO water and plants need it. That's the only thing special about this. And there's many others. So it's just that I use this because I'm using it on my orchids right now. I won't claim that that's the best fertilizer for either the orchids or the carnivorous plants. And there are many others available, and I group them in 2 groups. They react chemically slightly different. I recommend 20 ppm for Sarracenias, and that's because I think that's safe, because there was no salt buildup. Anything higher, you're on your own, and you have to watch for salt buildup. Also, if the higher you go, the more expensive it obviously gets. So this table here has the prices as of March, probably similar now on Amazon. So you can buy the MSU fertilizer in various package sizes online. There's at least 2 or 3 vendors who sell it. It all comes from the same company who makes it, Green Care, and they just package it into their own thing. So you can buy a ½ pound, a pound, 2 pounds, and 5 pounds. So if you use 20 ppm, even a ½ pound is 400 gallons. And then, of course, you know, if you buy 5 pounds, that's 4,000, more than 4,000 gallons. So that's a lot of water. So you pay, of course, more for small amounts because we pay shipping mostly. But if you have a small collection, there's no point in buying 5 pounds, because it's going to be taking you 20 years to go through that.
So everyone that you see who uses fertilizer, they usually recommend the vaccine.
Yes.
Now, the
And-
nitrogen is 16. Does that relate to can you relate that to the 20 parts per million or the 50 parts per million as far as?
You're asking a good question. That's all explained in my fertilizer presentation, but I will try to explain it here quickly. Let me just go into this table real quick. The final thing is the costs. I know everybody's cost conscious, but if you use RO water, even if you buy only ½ a pound, it adds 4 cents to a gallon of water. If you get your gallon from the vending machine, that's 35 to 50 cents last time I checked. So you might as well spend another 4 cents. And if you go more, you spend 1 cent if you buy 5 pounds. So fertilizer is dirt cheap. It's basically, considering how much plants cost and everything else, it doesn't need to be part of the equation. And this is the MSU fertilizer. Here's the same thing for a number of fertilizers that I picked and that other people use for carnivorous plants, including your Maxi. You can see the MSU fertilizer here is at the 5 pound is 1 cent per gallon. Some of the other fertilizers, unfortunately, you have to buy 25-pound bags, but if you're interested in them, I can bring you a pound. I have boatloads of fertilizer. This fertilizer amounts to 0.1 cent per gallon. So the MSU fertilizer is actually fairly expensive, and I buy 25-pound bags. It's probably one of the more expensive fertilizers. That's why I'm testing others right now and seeing, is it worth 10 times the or 5 times the cost. Here's some others that I recommend. So Peters Excel. Peters is, I think, the largest fertilizer manufacturer in the US. High quality fertilizers, and they're a little bit more expensive than Ultrasol. Ultrasol is this international conglomerate. I don't know where they're headquartered, but the fertilizer that the bag I have comes from Mexico. Whereas this is made in the US. Some people use Maxseed, and you can see Maxseed is 20 times more expensive than the MSU fertilizer. So it's, in my opinion, it's no. Fertilizer cost is not the main driver, but it is overpriced. And then someone asked me at the last meeting what I thought about the Super Thrive fertilizer, and I know Super Thrive for their little vitamin B drops. So I looked it up, and Super Thrive actually bought this company Dynagrow. I use Dynagrow in my orchids. Really good fertilizer, except it's liquid. So you're paying a lot of money to have the liquid shipped around the US. It's much more cost effective to use a powdered or granular. It's some of the Peters fertilizers are reground, so they're really a powder like sugar. The MSU fertilizer has different sizes of particles, smaller balls, tiny balls, more sandy-like stuff. I don't like that. I like the reground fertilizer, so it's more even. Oh yeah, the other thing is why I don't recommend buying huge bags. Fertilizer is hygroscopic. So if you take 10 years to use one up, you're going to have a concrete-like ball sitting in the bag, so which is really you need a hammer to break up. So smaller amounts are better. So now let me see if this is in here. And I may have ditched it for time. And if it's not in here, I ask, it should be. No. Oh, yeah, lots of experiments I don't need to go through that. That's something I just came up. I just want to mention I germinated 400 Sarracenias to do my next experiment. That was unintentional. I anticipated that only 10% or so would germinate. Pretty much I had 86 or so% germination rate. So I don't know what I will do with all these plants, but I have.
Did you document so you know your precision?
I actually haven't documented. So lots of stuff to do still. This was not all you, and if you're interested, I might have updates. So this is the end, and I'm ready for more fun. These are the 400 Sarracenia pictures. I might have to buy a second house to have another garage. Yeah, so it wasn't in here. So to go from the 13 or 12 or whatever percent to ppm, the two percentages don't relate to each other. Every good fertilizer should have a sheet, and actually Maxseed doesn't. The Peters fertilizer MSU, you go online, you type it in, and they tell you it's a long sheet, what's in there, what the elemental ratios, and then they tell you for 100 ppm dissolve X grams, 8 ounces, and they tell you for stock solution 100X, for an injection system, you dissolve this one. So every fertilizer should tell you how many grams you need to get 100 ppm N, or 20 or 50 in your final solution, and then you weigh this out, and then it also tells you that solution should have this EC. So after making it so that you don't have any errors in there, you measure it, and it's usually right on, give or take maybe 5%. So for a fertilizer that doesn't have that, use it on your lawn. No, really. A fertilizer should have a spec sheet that gives you everything, including pH and all of this, and all the reputable companies have that. So if some fertilizer doesn't have any like this, I would not touch it with a pole. No. Especially if it's 20 times as expensive as the next expensive fertilizer. Your choice. I mean, I know, I know. Now you imagine you have a 25-pound bag, right? Yes, oh, sorry.
I see that, the Plant Society has back pages, and I was just looking at one yesterday about fertilizer. And these are impressions, they're not studies. But I know some people were talking about how some success with seedlings, but once they got to a certain size, once rhizomes were formed, this is maybe another of those maybe wives' tales that needs to be spelled. But they were saying that before you get to a firm rhizome stage, you can throw lots of fertilizer at them, but you're actually generating conditions that can rot rhizomes with higher fertilizer. Have you encountered that?
No, you saw the pictures. And the weight, right? If I rot the rhizome out, the plant will rot and the weight would actually be reduced. No. So you saw how excited I was about statistical significant findings. I think with adult Sarracenias, it is extremely difficult to see significant results. I think to set up an experiment, and it makes it complicated because you have a rhizome that stores nutrients. So you have two equations, right? Nutrients drawn from the rhizome and you add nutrients. Plus, if they're in your yard, you have all sorts of, I mean, I don't have very many insects in my pitches, but I hear people that tell me their pitches are full of insects. So you have multiple conditions going on. I think that's experimentally impossible to untangle. That's why this under controlled condition in the garage is the best way of answering, because you're only asking one question and you answer that. With the adult plants, I'm taking some of the smaller ones that still fit in a terrarium, and I'm going to try the pitcher where there's no pitcher fertilization and see if I can tease out something. I'm not sure if I get to that because I think they will exceed the terrarium very quickly. I have hopes with these little guys that eventually, and I probably have a group where I don't put anything on the seedlings. So kind of like a sacrificial group, because without anything, they won't grow. Because no input, no output. But I'd like to document that, and then before they go down the drain, I can switch them back to fertilizer. Yeah, so my point is, try your own. It's fun. You learn something. And there's a ton of information on fertilizer. The ICPS, take it with a grain of salt. Because if you don't know precisely what they did, and if it's not documented, I wouldn't trust it. Yeah. John Britmacher or something. What? Yeah. Yeah, yeah. Now I remember the name. Okay, I hope you enjoyed it. And let me know if you want to hear the fertilizer, the intro into the kind of the chemistry and all of that. It's a bit complicated. So I warn you, I'm not going to give you a chemistry lesson, but I try to kind of summarize what's behind fertilizers. If there's any interest, more than happy to give that presentation. Okay, thank you. It was an hour. Thank you.
It's exciting.